Right. Now, after glycolysis in metabolism comes the transition stage. The transition stage. And remember, this is for aerobic metabolism. When you go on the aerobic pathway after glycolysis, you come to the transition or preparatory stage. Now, if we did not have any oxygen available, it would have to go over to the anaerobic stage and go through the fermentation process. But since we're talking about aerobic, meaning with oxygen, since we have oxygen available, it comes over here to the right to the transition or preparatory stage of the process and gets ready to go into Krebs. Now, we know glycolysis takes the initial glucose through the cell membrane and into the cell's cytoplasm, and then the rest of glycolysis is carried out in the cytoplasm of the cell. Well, like glycolysis, transition is going to take the pyruvate molecule through a membrane, but this time it's going to be the mitochondrial membrane because we're going to enter into the mitochondrial matrix to carry out the transition stage or preparatory phase of the process. So the transition stage, or also called pyruvate oxidation, or also called the preparatory phase of aerobic metabolism. It brings the two molecules, and remember, from now on, we're dealing with two molecules, not just one. So everything that we do, we have to take times two. So it brings the two molecules of pyruvate into the mitochondria through the mitochondrial membrane in here inside of the cell. Now, this is an oxygen-dependent process. It must have oxygen present. If it does not have oxygen present, it cannot do the aerobic metabolism and instead goes to the fermentation process, the fermentation pathway. So for every pyruvate molecule, as you can see here, this 3-carbon chain we start out with, and just keep in the back of your mind, we're doing 2 of these. For every pyruvate molecule we put in, we also, with this enzyme, pyruvate dehydrogenase, otherwise known as PDH. This is the enzyme that catalyzes this entire reaction. So you only have one enzyme for this whole thing. So what it does is it does 3 different individual actions. One is it takes that oxygen that we have to have present, and this is why we have to have oxygen present for this process to take effect. It takes the oxygen in the environment available in, and then it takes away one of the carbons in this 3-carbon chain of the pyruvate, and that turns it into CO2 instead of just oxygen, instead of just O2. It turns it into CO2 and it gives that off. So it takes off, the oxygen comes in, attaches to one of the carbons, and removes it by bonding the O2 to one of the C's, giving you CO2. And that gives us only a 2-carbon chain left after the removal of one of the carbons. But this enzyme also takes the coenzyme A, or just CoA for short, from already present in the mitochondrial matrix in the environment, and that takes that in. And so now it attaches that coenzyme A to the 2-carbon chain, and it takes in our good old friend NAD+, and that comes in, removes a hydride anion, the H negative, and that gives off our good old friend, the energy carrier NADH. And the result is this 2-carbon chain with a coenzyme A attached at the end, which is called acetyl-CoA. And that is the end result of this preparatory phase, this transition stage. So if we take this times the 2 pyruvate molecules we're going to be doing this to, for our net result, we're going to end up walking away with 2 CO2s and 2 NADHs, and then of course our 2 acetyl-CoAs at the very end.